Now, enzymes. Enzymes, they are catalysts. Number of chemical reaction takes place in an organism due to the enzyme, with the help of the enzyme. Enzymes are the catalyst for all the organic chemical reaction which are taking place in our body. Now, enzymes are protein. Some enzymes are nucleic acid which are known as ribozymes. Now, proteins, they are primary structure, they are long thread-like structure, they become secondary structure by twisting, becoming in helix, and they twist more to form tertiary structure. Now, as the backbone of the protein is forming the tertiary structure, it is folding, it is crisscrossing each other. So, a pocket is formed. Crevices are formed. There is empty space in between. Now that space becomes the active site for the substrate. For breaking or making, enzymes are required. So whatever can be a substrate, that will get fixed in the active site. And whatever the reaction has to be taken place, whether it is to be broken or made into one, that takes place in the active site. So the substrate fixes in the active site and then it comes out as a product. Now, the enzymes, they perform function at a particular temperature. And for normal organism, it is 40 degrees centigrade. Above that, it gets degraded. It's not able to perform the functions. But there are some thermophilic bacteria organisms which can perform function at 80 to 90 degrees centigrade. Their enzymes are active, they are not degraded, they are able to perform function at 90 degrees centigrade also. These are the enzymes, the organic catalyst. But the inorganic catalyst, they are able to perform at very high temperature and pressure. That is the difference between organic and inorganic catalyst. Inorganic catalysts, they will perform best at high temperature and pressure, whereas the organic catalyst, these enzymes, they will perform best at a particular temperature. That is around 40 degrees centigrade. So here enzymes are catalysts, they are protein, they have primary structure, some nucleic acid, they perform the function of enzymes, they are known as ribozymes. Now, they form tertiary structure, so the backbone of the protein chain folds upon itself, crisscrosses itself, many crevices are formed, pockets are formed, which forms the active site. Substrate fixes in this active site. So, they catalyze the reaction at a very high rate. There are thermophilic organisms which perform best at 80 to 90 degree centigrade, but normal enzymes, they get paralyzed above 40 degree centigrade. Now the reactions are of two types, physical and chemical. In physical, there is only change in the state takes place. You take an ice cube, you heat it, the distance between the molecule increases to form water, and you still further heat it, distance between molecule increases and changes the state to water vapor. But in chemical reaction, the breaking and forming of bond takes place, whether it's an organic or inorganic, it is all in chemical reaction, the breaking and forming of bond takes place. Now, if barium hydroxide reacts with sulfuric acid. It forms barium sulfate and water molecule is formed. So this is a chemical reaction. The bond has broken and new bonds have been formed to form new product. Now, in the organic reaction, hydrolysis of starch takes place to form glucose. This is also a chemical reaction. So these are the chemical reactions. The breaking and forming of bond has taken place, and there is a certain rate of reaction. The rate of reaction is at what speed it takes place, at what speed, in how much time the product is formed. So here rate is delta P upon delta T, that is the amount of product formed per unit time. Now if the direction is given, then you can find the velocity also of the chemical reaction. Now when you heat, the chemical rate increases. It doubles at every 10 degrees centigrade rise in temperature, and vice versa. If you reduce, then the rate becomes half. So at every 10 degrees centigrade, the rate of reaction doubles. Now, but in the organic chemistry, in the organic biochemistry, the enzymes, which are the catalyst, which increases the rate of reaction, they get destroyed at very high temperature. Now you take a simple example of carbon dioxide and water vapor. Carbon dioxide will dissolve in water. It will dissolve 200 molecules per hour without any help. It will dissolve. But this enzyme carbonic anhydrase is present in the cytoplasm, which will help carbon dioxide to dissolve in water. So this is the enzyme which helps the reaction to take place. It will form carbonic acid. Without this enzyme, carbon dioxide will dissolve in water to form carbonic acid, but in cytoplasm, with the help of carbonic anhydrase, the rate will increase. The rate will increase. 6 lakh molecules will be formed per second. So the rate of reaction has increased by 10 million times. So this is the power of catalyst. This is the power of the enzymes, which speeds up the reaction and increases the rate to a very quick. So the power of enzyme is incredible. Now there are thousands of enzymes which are for different reactions, for metabolic reactions. Thousands of enzymes present in our system for the metabolic pathway. Now here a multi-step chemical reaction, when each step is catalyzed by the same enzyme complex or different enzyme, is called the metabolic pathway. So the metabolic pathway will have number of enzymes. Some reaction will be carried by the same enzyme or might be different. So various enzymes, they react, they help the reaction to take place at a faster rate in the metabolic pathway. The example here is glucose is going to form pyruvic acid, is going to produce energy. It takes place in 10 steps. 10 enzymes are required for glucose to convert into pyruvic acid and energy is released. So 10 enzymes are necessary for this metabolic pathway. C6H12O6 plus oxygen, it gives 2 molecules of C3H4O3 and 2 molecules of water. Now, one or two steps when added in this metabolic reaction, the product might be different. Now, under anaerobic condition, the skeletal muscles will produce lactic acid. Now, here glucose is producing pyruvic acid under aerobic condition. Now skeletal muscle under anaerobic, now here the condition has changed, it become anaerobic condition, it will produce lactic acid. So when you have to do some extra work, then your muscles will produce lactic acid and you will have pain in your body. Now yeast, idly batter yeast, they are multiplying, they undergo anaerobic respiration, and you ferment the yeast, ferment the idli batter with the help of yeast. So they undergo fermentation, the end product is ethanol. So here in different condition, different products are formed. Now, the chemical reaction or metabolism, the metabolic changes, the biomolecules, they change. Now, they change with the help of enzymes. Enzymes are protein, tertiary in structure. They have crevices in between. Now, the substance, the molecules, they are embedded in the crevices. That is known as the active site. Now, the chemical which has to change, that is known as substrate. They get embedded in the enzyme at the active site. So now whatever the chemical change has to take place, whether the bond has to form or has to be broken, that takes place in the active site. Now after the change, what we get is the product. We might get two product or two combined into one, whatever, we get the product. Now to form the product, the substrate has to fix with the enzyme and they form a complex which is known as ES. ES is a complex of enzyme and substrate. They are undergoing change to form the product. So now this state where the changes are taking place is known as transient phenomena. That is, the changes are taking place in the substrate with the help of enzyme, and then you get the product. Now from the substrate, it gets attached to the enzyme. This is transient phenomena. There are many altered structural stages in between which may take place. It might be one structural change or many structural changes which are taking place. Now, after you get the product, the chemical reaction is over, but you have to understand what happens in between. What is the role of the enzyme? So this diagram shows the state of the substrate. This y-axis shows the potential energy of the substrate. The substrate is the substance where the chemical reaction is taking place. So it shows the potential energy of the substrate. Now as it gets converted into product, there is a transitional state which takes place. Without the enzyme, the time taken for this transition state is more. With the help of enzyme, this transition state is shortened. So here the activation energy without enzyme is more. Activation energy with the enzyme is less. So it changes to product very quickly. It might be a exothermic reaction, it might be endothermic reaction, whatever the transition takes place, and there is a definite energy which is present in the substrate, that is known as the activation energy. So what the enzyme does, the activation energy is reduced from potential energy, it comes to the product which is the stable state which does not have any energy. The energy is given up, it becomes stable. product is stable. So here this transition energy changes into product. Without enzyme, the transition energy is more, so it has to undergo, a substrate has to undergo transition state. It has to, this is the transition state here. Now this is the transition state here. This is the activation energy. This is the activation energy, and it becomes a product which is stable. So it becomes stable. Now, difference in average energy content of substrate from the transition state is called the activation energy. So, this is the activation energy. So, this graph shows the concept of activation energy, the potential energy of the substrate, it combining with the enzyme in the getting embedded in the active site, the reaction taking place, and it is getting converted into product. It might join or it might break. Whatever the enzyme is responsible for, the reaction will take place and you get a product. So there is a transition state before getting the end product, that is the complex enzyme substrate complex is formed, which is the transition state, transient phenomena, and there is an altered structural state which is taking place, which might be in 1 or 2, 3 steps. So now there is a difference in energy from this initial substrate to the product. It becomes stable here. So in between there is a change in energy which is known as the activation energy. Now the nature of the enzyme reaction. Enzyme is a catalyst. It helps the reaction to take place at a fast rate but does not take part in the chemical reaction. It's a catalyst. Now, enzyme has got an active site in which the substrate, the chemical, the molecule on which the reaction has to take place, it fixes in the active site of the enzyme. It's a crevice, it's a space in between active sites, so substrate is fixed. As soon as the substrate is fixed, the enzyme tightens its hold, and now it has formed a complex. It's a enzyme substrate complex have been formed, and the chemical bond of the substrate is broken by the enzyme. Now this enzyme substrate complex turns into enzyme product complex. New substances form. After the enzyme product complex, the enzymes gives out the product, and now enzyme is free to bind with another substrate. This is how enzyme works. So now the nature of enzyme action, enzyme plus substrate, it forms ES and then converts into EP and then forms enzyme and product. Product is what we want. Enzyme is again going to take substrate, convert into product. So the substrate binds to the active site. Binding of the substrate induces the enzyme to alter its shapes, to fix the substrate in it. Enzyme alters the shape. Active site breaks the chemical bond of the substrate. New enzyme product complexes form. The enzyme releases the product of the reaction. Free enzyme is ready to bind to another molecule. Now, factors affecting the enzyme activity. Now, the enzyme activity can be affected under certain conditions because the tertiary structure of the protein is altered. Now, the pH and the temperature are the conditions which affects the enzyme activity. Now, the pH has to be optimum for the enzyme activity to take place. It will go on increasing with the pH until the maximum activity will take place at an optimum pH, and otherwise it goes on decreasing. It is slow before that optimum pH, and it declines after the optimum pH. So maximum activity is at the optimum value of that pH. Now even for the temperature in the low temperature, the enzymes, their activities slow down. They are not active, but at very high temperature the protein get denatures. It is not able to work at all. So temperature, there has to be an optimum temperature for the organism. It is normally 40 degrees centigrade, which is the optimum temperature for the enzyme activity to take place. So the pH and temperature has to be optimum for maximum enzyme activity to take place. Now, even the substrate affects the activity, the velocity, the rate of the chemical reaction. Now, as you increase the substrate, the enzyme activity goes on increasing, and it reaches a state where it becomes maximum. Why? Because all the enzymes, they are occupied now. They have substrate filled up. After this state where all the enzymes are occupied, no more increase in the velocity of reaction will take place. So that is known as Vmax. Maximum velocity will be reached. So no more increase in the velocity of the reaction takes place. Why? Because all the enzymes are occupied with the substrate. Now you go on adding substrate. There's no enzyme for the reaction to take place. So here it has reached maximum. This is V by 2 max. Now, so therefore the concentration of substrate will increase the reaction to a certain rate, and then the rate is constant. Now, in nature, whatever the substrate, it is changed by the enzyme. It has to be stop. So inhibitors, they stop. They have chemical structure similar to the substrate. So they fix in the enzyme and they stop the reaction. So they are known as inhibitors, and since their structure is similar to the substrates, they are known as competitive inhibitors. Inhibitors, they resemble the substrate. Now succinic dehydrogenase, their production is stopped by malonate because its structure is similar to the succinic dehydrogenase. So these are the factors which affect the enzyme activities, that is pH, temperature, and the concentration of the substrate in the reaction. Competitive inhibitors are often used in the control of bacterial pathogens. Now, the classification and nomenclature of enzyme. There are thousands of enzymes which are isolated, studied, grouped, depending upon the catalytic action it does. So there are 6 groups. They are classified according to the functions. They are classified into 6 groups, which are further divided into subclasses, 4 to 13 subclasses. So let us see which are the types of enzymes. Oxidoreductases, that is, it will reduce one substrate and oxidize the other substrate. So here the substrate is reduced and this substrate is oxidized. Now this another first one which was reduced is oxidized and the second one is reduced. So oxidoreductases, they do the function of oxidizing and reducing. transferases, they just transfer one group from one substrate to other. So the group was attached to the first substrate, now it is attached in the, after the product formation, the group is attached to the second substrate. So transferases, they just transfer the group. Hydrolysis takes place. Water is removed. Enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic, carbon halide, phosphorus nitrogen bonds. So hydrolysis takes place with the help of hydrolysis. Lysis, they remove not water but the group and forms double bond. So enzymes that catalyze removal of groups from the substrate by mechanism other than hydrolysis, leaving double bond. So here X is attached, it will be removed. So Y is attached, Y is removed, and then carbon double bond is formed. Now isomerase, it catalyzes the interconversion of optical, geometrical or positional isomers. So it changes the isomers. Ligases, that is catalyzing the linking together of two compounds, and it forms bond between carbon, hydrogen, carbon, sulfur, carbon, nitrogen, phosphorus, oxygen, etc. bonds. Now the cofactors play a very crucial role in the catalytic activity along with the enzymes. Enzymes, they are polypeptide chain, they are protein part, that is apoenzyme. Now along with this protein part, the apoenzyme are the non-protein part, that is the cofactor. They help in catalytic activity, they help the enzyme to perform the function of catalysis. Now these cofactors are of three types. They are attached to the apoenzymes. Prosthetic, coenzyme and metal ions are the three types of the cofactors. Prosthetic, coenzyme, metal ions are the three types of cofactors. Now this prosthetic and the coenzymes are the organic compounds. Both are organic compounds. Here prosthetic, they are tightly bound to the apoenzymes. Apoenzymes are the protein portion. They are very tightly attached to the protein part. Now the example in the case of hydrogen peroxide. Hydrogen peroxide, it requires the enzyme peroxidase and catalase to form hydrogen oxygen. So here heme is the prosthetic group. It gets attached to the active site of the enzyme. So this is the example of separation of hydrogen and oxygen in hydrogen peroxide, which takes place with the help of peroxidases and catalases, and heme is the prosthetic group attached to the active site. So they are tightly bound to the apoenzyme. Now coenzymes are also cofactor, but they are transient. Then coenzymes, they help many enzymatic actions. Here prosthetic, they help only in one type, but one coenzyme will help many enzymatic actions, so they are transient. They are attached only for while the catalysis is taking place. And here they require vitamins. Vitamins are required in the coenzymes. Now the example here is nicotinamide adenine dinucleotide. They require the vitamin niacin. Now, here metal ions, they are also cofactors. They have to be present along with the apoenzyme for the reaction, for the action of the enzyme to take place. What the metal ions do is they form a bond, they form a coordination bond with the side chain on the active side. On the side chain, they form the coordination bond, and they also form coordination bond with the substrate. So they form coordination bond with the enzyme, with the apoenzyme, and they form the coordination chain with the substrate. This is how they form a chain, and they help the enzymatic action catalysis to take place. So this is how the metal ions they work. So here cofactors are very important for catalytic activity to take place, and without the cofactors the enzymes do not perform the catalysis. So cofactors are must for the enzymes to perform the function of catalysis. The protein part is apoenzyme. They are polypeptide chains, and non-protein, they are cofactors, prosthetic, coenzymes, and metal ions are the three cofactors. So cofactors are very important for enzymatic action. Zinc is a cofactor for the proteolytic enzyme carboxypeptidase.